This is the third and the last video for cell physiology, and in it we'll finish up talking about cytoplasmic organelles and inclusions. The only thing we'll have left to do for cell physiology is cell replication and mitosis, which we'll cover in class. The next organelle that we're going to talk about is also phospholipid bilayer enclosed, and it is the smooth endoplasmic reticulum. What you see here is a very common depiction of the smooth ER, but it's not a very good one. And so if you see these in books or online, you should know that it doesn't do a good job of showing you what the smooth ER looks like. This is a much better picture. As I said, the smooth ER is made of phospholipid bilayer, and it is phospholipid bilayer which arises from the rough endoplasmic reticulum. So in general, smooth endoplasmic reticulum is a little bit further away from the nucleus than the rough endoplasmic reticulum. Again, just let me remind you that the rough endoplasmic reticulum arises from the nuclear envelope. Another thing that happens with the smooth ER is that the flattened tubules morph into circular tubules. And so here you can see in cross section, under the microscope, these just look like little circles. And so there you can see that. The third difference is that, of course, the smooth ER doesn't have any ribosomes along it the way that the rough ER is, hence the name smooth endoplasmic reticulum. The job of the smooth endoplasmic reticulum, like that of rough endoplasmic reticulum, is to synthesize biomolecules. But in this case, what's being synthesized would be lipids rather than proteins. And in particular, the lipids that are synthesized include fatty acids, phospholipids, and steroids, which might in fact be steroid hormones, things like testosterone, cortisol, estrogen, those types of things. Another job of smooth endoplasmic reticulum is to metabolize carbohydrates, and we see this in particular in the hepatocytes. You know that the liver has the job of trying to help keep your blood glucose up when you haven't eaten for a while, and the smooth endoplasmic reticulum helps by breaking glycogen down into glucose, which the hepatocytes can then release into your bloodstream. The smooth endoplasmic reticulum also helps to neutralize drugs and toxic chemicals, and this primarily takes place in the liver, but also in the kidneys. The drugs that are being neutralized could be medications that you've been prescribed or not, or they could be recreational drugs. Alcohol also is a toxic chemical, which is neutralized by smooth endoplasmic reticulum, and again, particularly in the liver. And so it wouldn't be surprising that on any given weekend, the smooth endoplasmic reticulum of DePauw students is pretty busy trying to help neutralize some toxic chemicals. The endoplasmic reticulum that we find in skeletal and cardiac muscle is specialized to sequester and release calcium. And we'll talk later about why that is and what that does for muscle function. But because of that, we give the muscle endoplasmic reticulum a special name, and we call it sarcoplasmic reticulum. Now, most of you know that the prefix myo means muscle. Well, there's a second prefix which means muscle, and obviously that is sarco. So again, the skeletal muscle and cardiac muscle endoplasmic reticulum is referred to as sarcoplasmic reticulum. And here's a really nice depiction of the entire smooth endoplasmic reticulum. And again, you see it arising from the rough endoplasmic reticulum. Again, there are no ribosomes here. The ribosomes are these small yellowish dots that you see there, but you don't see any on the smooth endoplasmic reticulum. The tubules are circular in nature, and they're not flattened sacs. And then lastly, this does indeed look as if it's somewhat net-like, and that's what the word reticulum means. And so that's why endoplasmic reticulum is given that name, because it is somewhat net-like. The next membrane-bound organelle that we're going to talk about is the Golgi apparatus, and many people pronounce this Golgi, but technically it's Golgi. It's also referred to as the Golgi complex, and in some older books, Golgi body. The jobs of the Golgi apparatus are to, number one, modify the molecules that it receives from rough and smooth endoplasmic reticulum. And you can see some of those packages coming in here from the rough endoplasmic reticulum delivering protein. Well, that protein will then get modified. In some cases, we take off sequences of amino acids and end up with a shorter protein. In other cases, we clip it up into segments and rearrange them completely. And then another thing that can happen is we can actually add carbohydrate to the protein and make it into glycoproteins or proteoglycans. And we talked about those when we talked about chemistry and also when we talked about the structure of the cell membrane. The Golgi apparatus can also take in lipids coming from the smooth endoplasmic reticulum, and it can modify those as well. And again, one of the things that can happen is we can add carbohydrate to make glycolipids. After those have been modified, they get packaged up at the other end and then sent out to some other place. So first of all, you might look at this and think about the location of the Golgi apparatus. So pause the video for a second and explain where it's located. And so what you should have said is that the Golgi apparatus is receiving things from the endoplasmic reticulum, so it's further away from the nucleus than the endoplasmic reticulum, but it sends stuff out to the plasma membrane, and so we can see that it's in between the ER and the plasma membrane. This picture shows you a little bit more complete depiction of Golgi apparatus. And here again, just to reiterate, it is a stack of membranous, in other words, phospholipid bilayer flattened sacs. And so right along here, it'd be nice if they did this in a different color. This is phospholipid bilayer. And it almost looks a bit like nuclear envelope, but it isn't. There is nothing like the nuclear pore complex that connects these two membranes that you see here. And then in this picture, you can see that these look somewhat like shallow bowls nested inside one another, and it shows 4 of them. But typically what we find is somewhere around 5 to 8 of these shallow nesting bowls in the Golgi apparatus. And then finally, here's a real nice picture. First of all, what we have up at the top is a transmission electron micrograph of Golgi apparatus that we've seen for years. And it's hard to really get a good idea of the structure of this thing from just this two-dimensional picture. Well, somebody decided what they would do is they would draw in the Golgi apparatus underneath the transmission electron micrograph. And so this reddish rust-colored thing that you see is the Golgi apparatus. And again, just to remind you, you can see here we've got phospholipid bilayer, and like the Ruffiar, they're kind of flattened sacs. The other thing that this is showing you is these little phospholipid bilayer vesicles that are arriving from the endoplasmic reticulum. Because both of these are phospholipid, and because of the fluid nature of that, they can merge. The chemical that's in here gets released to the inside where, again, it will be modified, and then it gets packaged up at the other end and sent out for whatever job it is that it has. And so here you can see kind of a better overview of all of that. Here's the nucleus, nuclear envelope. Here's the rough endoplasmic reticulum. Presumably this is smooth endoplasmic reticulum, and so it should be synthesizing lipid, but they're telling us that it's protein. So I think that they've made a mistake here. Either this should be dotted with ribosomes, or they should tell us that it's lipid. But at any rate, here you can see that phospholipid vesicle that has pinched off from there. It moves to the Golgi apparatus. Whatever the molecule is modified and then repackaged and then sent out. And here you can see a slightly different depiction of that. Here we just simply see the Golgi apparatus, but the blue vesicles that you see are coming from the endoplasmic reticulum. And since they're saying it's rough endoplasmic reticulum, you know that the molecule inside of there has to be protein. But again, it's going to get modified. They don't really show you the modifications that are taking place here in the Golgi apparatus, but through the semester and next semester, we'll talk more detail about some of those modifications. And then finally, the molecule that we end up with is repackaged and sent off to be used for some specific task. And so one thing that may happen is that we may be making a molecule that's going to be released to the outside of the cell through a process of exocytosis. And so if this is protein, it could be neurotransmitter that's released and goes to another nerve cell. It could be hormone, which has an impact on other cells of the body, or it could be something else, but it's being released to the outside via exocytosis. The other thing that could happen is that the protein that's here could actually already be incorporated into the membrane of the vesicle, and then this membrane becomes part of the plasma membrane, and so now those proteins have also become a part of the plasma membrane. And they could be transport proteins or channels to allow stuff to either move into or out of the cell. They could be receptors, or they could even be enzymes. And then lastly, sometimes this protein stays inside the cell and is used there, as is the case when we make a lysosome, which is what we're going to talk about next. A lysosome is a very, very, very small organelle, much, much smaller than the others that we've talked about, but it is also a phospholipid sac. And so here you can see the phospholipids. The yellow would be the phosphate head, and the green would be the lipid tails. And here you can see a lysosome. Again, remember that it's three-dimensional, not two-dimensional, as you see here. A substance is brought into the cell. In this case, it looks like endocytosis. It could be pinocytosis, but once it's been brought in, this vesicle merges with the lysosome. Again, remember, fluid nature of the phospholipid bilayer. And the hydrolytic enzymes that we find in there will now attack the covalent bonds of whatever this is and break it down into smaller substances. And so again, as we look at this, it contains lots of hydrolytic enzymes. Hydrolytic enzymes are going to do what? And I hope you immediately said they're going to cause hydrolysis, or breaking of covalent bonds by adding water to the bond. The lysosomes also have a low pH, which also help them to function. For one thing, if they're working to break down protein, it can help to denature it. For this reason, the lysosome is sometimes referred to as the digestive system of the cell. And it can digest a number of things. For one thing, it can actually digest worn out cells that are brought into a phagocyte, or it can digest organelles that need to be replaced. They're old, they're worn out, they don't function anymore. Well, you know that phage means to eat, and auto is self. So autophagia is this eating of its own things, own cells, own organelles. It can also break down glycogen granules to get glucose to be used in cellular respiration, or it can break down lipid droplets to also help with energy metabolism. And then again, as we said earlier, these red dots that you see up here could in fact be foreign material. So it could be something like a bacteria or something along those lines, which could cause infection and is taken into the cell by either phagocytosis or endocytosis. And then again, the lysosome can help to digest that and break it down. And then another important function of lysosome is it helps with bone remodeling. Even though most of you are probably full grown now, you're going to continue to remodel your bones for the rest of your life. And approximately every 5 years, you'll have a brand new skeleton. And that's about how long it takes to break down and replace all the bone in your entire skeleton. But the lysosomes in cells called osteoclasts are very important in helping with that bone breakdown so that you can rebuild it. And here we can see a demonstration of phagocytosis. And here is the phagocytic cell. So it could be a macrophage, a big eater, or it could be a neutrophil. At any rate, what it's doing is it's reaching out and engulfing this cell or this particle. Once that is inside, then the lysosome will merge with it, and the hydrolytic enzymes will break it down so that we can get rid of it. I do want to point out to you, sometimes people make the mistake of thinking that the lysosome is a cell. It's not. The lysosome is one of the organelles, and organelles and inclusions are parts of cells. They are not cells themselves. And it's not the lysosome which does phagocytosis. It's the cell itself. A lysosome then helps to digest what has been brought into the cell, but it's not the lysosome which is doing phagocytosis. And it appears that the lysosomes are actually made from the ER and then will take in proteins that were made in the ER and modified in the Golgi apparatus. Here you can see a number of the membrane-bound organelles that work together, and they work together in what's been referred to as an endomembrane system or an endomembrane complex. And what I would like you to do is to pause the video, and I would like you to identify those things in the cell which are enclosed in phospholipid bilayer, and then talk a little bit, write down how they interact with one another to do their jobs. Okay, if you want, you can bring that to me and I could take a look at it and talk to you about it and tell you whether or not you're missing anything. An organelle, which is very similar to a lysosome, but is much, much, much smaller, is a peroxisome. And so a peroxisome, too, is a little vesicle which is encased in phospholipid bilayer, and it contains lots and lots of enzymes as well. The peroxisome actually provides a compartment for oxidation reactions. And remember, we talked about oxidation reactions when we did chemistry. And in particular, things that get oxidized include long-chain fatty acids and also amino acids. One of the problems with this oxidation of these molecules is that this results in the production of H2O2. And many of you may remember what H2O2 is. You might pause and see if you can identify it. The production of H2O2 from oxidation of things is what gives the organelle its name. And so there's a hint for you if you're not sure what H2O2 is. Well, H2O2 is actually hydrogen peroxide. And again, it's toxic to the tissues of your body. And so another enzyme that we find inside the peroxisome is catalase. And catalase will take two hydrogen peroxides, two H2O2s, and will convert them to water and oxygen, which aren't toxic to us. It turns out that the peroxisome has another job. It also plays a role in synthesis of lipids, much like the smooth endoplasmic reticulum does. And so it can produce cholesterol and cholesterol-like molecules. It can produce phospholipids. These phospholipids tend to be important in production of the myelin sheath that we find around axons of neurons. And we'll talk about myelin later when we get to the nervous system. And then a cholesterol-related substance is bile acids, and these are needed to digest fats that you eat in your diet. And so again, the peroxisome will break things down, but it also plays a role in synthesis. The latest research suggests that peroxisomes are actually made from vesicles that come from the endoplasmic reticulum and the mitochondria. And those merge to make a bigger vesicle, which then can split in half to make two peroxisomes. And again, I want to reiterate, peroxisomes, very small, much, much, much smaller than the lysosomes. And then the last membrane-bound organelle that we're going to look at is the mitochondria. And so here you can see both a drawing of a mitochondria and also a photomicrograph of a mitochondria. And here's another drawing of a mitochondria. And you already know that the mitochondria is known as the powerhouse of the cell, and that's because it's the site of cellular respiration, which is where we make the majority of our ATP. And I do want you guys to know the overall reaction for cellular respiration. And if you're kinesiology students, I ask you this all the time, and I want you to be able to tell me what it is. C6H12O6 is often identified as glucose, and yes, that's the correct formula for glucose, but this is also the formula for fructose and galactose. Those are all isomers, and so they're very similar. And that glucose in the presence of oxygen is broken down to CO2 and water. And in the meantime, we also make ATP as we're doing this. And so cellular respiration is using oxygen as the final electron acceptor to release energy from the foodstuffs that you've eaten, carbohydrate, fat, and protein. Now, what I'd like you to do is I'd like you to look at this mitochondria, pause the video, and identify the important parts that play a role in cellular respiration. And what you should have said is that the important parts include an outer membrane, which is indeed phospholipid bilayer. And here you can see it. It's dark brown, so it stands out well from every other part of the mitochondria. There's also an inner membrane, which here is kind of a beige or a pale tan. This is also phospholipid bilayer. And so again, you might think, well, it's a little bit like the nuclear envelope. But again, you can see that there is nothing like a nuclear pore complex which connects these two membranes. Here you can see blue, and just like people who make maps show water lakes, rivers, oceans in blue, they're showing you a solution with a watery solvent in blue as well. But it's much, much, much more than that because it is a solution, lots and lots and lots of chemicals in there, including lots of enzymes. This is what's known as the mitochondrial matrix. You don't see it quite as well up here. They don't show it standing out quite as much. And then the fourth important part that you need to know about is this space that we find between the outer membrane and the inner membrane. And this is in between, and so we use the prefix inter, meaning in between, intermembrane space. And so those are the four parts of the mitochondria that I want you to know about: outer membrane, inner membrane, matrix, and intermembrane space. And I really don't care about these folds, which are listed as cristae. As you might have guessed, the importance of those is they help to increase the surface area of the mitochondrion. And now we come to our last cytoplasmic organelle, which is not membrane bound. And oftentimes books, teachers, websites identify this wrong. They talk about centrioles, and it's not the centriole, which is the organelle here. It is the centrosome. The centriole is a part of the organelle, and all of you know the centriole is this two little spindles that you see here that generally are shown at right angles to one another. The centrosome is the main microtubule organizing center. Remember, microtubules are one of the three things that make up the cytoskeleton of the cell. And for this reason, the centrosome is known by some people as the MTOC, microtubule organizing center. The two parts of it are the two centrioles that we just mentioned. And then if you look very carefully here, you see kind of this bright violet around the outside of the centriole. That's what's known as paracentriolar material. And here they've shown it to you as this kind of cloudy, amorphous, bluish purplish stuff. And so that's the paracentriolar material, and here is one centriole, here's the other centriole. The interesting thing is that the centrosome is required for the survival of the cell, but the centriole is not. They actually have techniques where they can go in and they can pluck out the centriole, leaving behind the centrosome, and the cell will survive. However, if they remove the centrosome, the cell won't survive, it dies. The centrosome and the centrioles are very important, and they replicate during the S phase of mitosis. And again, in class, we'll talk about mitosis and go through the different phases of it. The centrosome is responsible for creating these astral microtubules during mitosis. And again, those of you who've had a fair amount of biology or remember your high school biology really well, you know these microtubules attach to the chromosomes and pull them to opposite poles as we're replicating the nucleus and the cell. And so now we've covered all our organelles and inclusions, and what I'd like you to do is think about what this means for the structure of a cell when we talk about what a cell is specialized to do. So if a cell is specialized to synthesize and secrete protein, what would you expect to find when you look at that cell under a microscope? Pause the video and give some ideas. If you said you expected to see lots of rough endoplasmic reticulum, well, you'd be right, because that's the job of rough endoplasmic reticulum, make protein. Well, making protein is a highly energetic process. That's an example of chemical work, and we need a lot of ATP. So in addition, we're going to have a lot of mitochondria here, and you can see that there are quite a few mitochondria in this cell to go along with rough endoplasmic reticulum. What would you expect to find in a cell that specializes in actively transporting substances into a cell? And your hint here is the term active. And so if you said you'd expect to find a lot of mitochondria because we need ATP to support the transport work being done, then you're correct. What about a cell that's specialized to synthesize lipids? Well, you'd expect to find a lot of smooth endoplasmic reticulum. And here you can see the smooth endoplasmic reticulum cut in cross-section. And so again, it just looks like these circles or these holes. Again, this is the edge of those tubules, those phospholipid tubules that we see. This is actually a picture from cells of the testes, which are responsible for making testosterone. Testosterone is made from cholesterol, which indeed is a lipid. So not surprising that we find a lot of smooth endoplasmic reticulum there. And then finally, what would you expect to find in a cell which phagocytizes lots and lots of foreign particles? Well, remembering that we have to digest those things once they're taken into the cell, you should have said, oh, I expect to see a lot of lysosomes, because then we're going to have to release lots of hydrolytic enzyme to break down and digest whatever it is that we've brought into the inside of the cell. And so that brings us to the end of our cell physiology videos. And again, in class, we'll start the next PowerPoint, probably not next class, but the class after that, and go over cell replication and mitosis.